Let me show you one flower. I think screen is visible to you.
Yes, sir.
Three I have drawn. There will be two more on the other side. Which flower it is?
P.
So similar flower you have seen. You have drawn also earlier. These are two wings, and the innermost is what?
E.
Kid. Have you seen that flower?
So white one I have seen.
Hmm? White one.
So
Okay,
white.
whatever it may be. You've seen the white color. So if you have seen this flower, have you seen when you normally see the flower? Have you seen any gynoecium, androecium, all those things?
No, sir.
No, why? Why they remain
Inside there.
inside the where? Inside the keel. Is that clear? So because keel is not opening properly, so it is closed. Is this clear?
Yes, sir.
So what happens? You may see at towards maturity, you may find this keel is little bit up to this much, it is opening, right? If keel opens up to this much, at that time you can see this uh, um, the stigma and the anther part of the stamen, right? So at that time already it is pollination has already occurred, because pollination occurs in bud condition. Pollination occur when it is in bud condition. Are you getting my point?
Yes, sir.
So what happens? Pollination, this plant is complete self-pollinating flower. Okay, they have self-pollinating flower, and uh, during bud condition already pollination has taken place. So afterwards, if it blooms also, this part, if afterward bloom also, then also it is having no function. No, it cannot attract cross-pollination. Is this clear? So this particular plant means in case of pea plant, what we find?
Self-pollination.
Self-pollination. I think your confusion has gone.
Sir,
Hmm.
so in F1 generation cross selfing is done. So that is self-pollination only, no?
Self-pollination only. Selfing means self-pollination only.
Okay,
That's why
sir.
selfing is possible in case of plants, not in case of animals. Are you getting my point? Can you go for selfing in dogs or cats or human beings? Is it possible?
No.
No. So if you are asked that why not this Mendelian this process are possible in case of human being, that F1 generation from F1 to F2 generation if you want, then answer will be what? We cannot go for selfing of F1 generation in humans or in any animals. This is possible only in case the organism is bisexual. Is it clear?
Yes, sir. Sir,
Hmm.
genetics is called the study of heredity. So heredity is
Heredity and variation. Heredity and variation.
both.
Both.
So heredity means only similarities.
Heredity, the characters what is inherited. So characters which is inherited are similarities only. Variation means if the characters are inherited, then why variation appears? Question is that. Actually, variations all of a sudden you are not getting abrupt variation. We find a continuous variation. That means what? Variation, say original combination what was in the father, we are not getting the next generation. Rather, we are getting a mixture of both the parents. So that leads to variation. That is a kind of what? Recombination. Is not it?
Yes, sir.
Okay. So this type of variations are actually continuous variation, not discontinuous variation as in case of mutation. When mutation occur, all of a sudden a dramatic change will occur. Is not it? So that variation is saltatory or mutation.
Okay, sir.
So we have discussed up to which portion?
The third conclusion.
Third conclusion. That means we have discussed only monohybrid cross. What is the monohybrid cross?
When only one character is concerned to us while doing the cross.
Okay, when we consider only one character in our consideration. So that is monohybrid cross. Now, if we go for Aja, what is the test? Is what in a monohybrid cross? What is the phenotypic and genotypic ratio?
Phenotypic 3:1. Genotypic 1:2:1.
2:1.
Phenotypic is what?
3:1.
3:1.
Why 3:1?
Three were tall and one
Three
was
were
Tall,
tall
one
and
dwarf.
one dwarf. Okay.
One
And
dwarf.
genotypic?
1:2:1.
1:2:1.
Why it is so?
So one
one
homozygous tall, two heterozygous tall, and one
Homozygous dwarf.
dwarf.
Okay, very good. So the ratio you have got like this. So remember these values. Now let us go for dihybrid cross. So when you say dihybrid cross, acha, I'll go for dihybrid cross. But before that, some very important point you should know. Tell me first, what are the 7 characters Mendel has chosen?
seed color, seed shape, pot color,
Seed
pot
color,
shape,
seed
stem
color,
height,
flower color,
flower position, and
Seed color,
seed
the seed shape,
seed shape,
the
okay.
pot
Another
shape, pot
pot
color,
color, pot color, pot
pot
color, shape,
color,
Flower color.
pot shape,
pot
Pot shape.
flower
shape,
color, flower
flower
position,
color,
and stem height.
flower color,
Stem height.
flower position,
flower position, and
stem
stem
height.
Stem
height.
height.
Okay, very good. Now, again, where we may get confused, that is which one is dominant and which character is a recessive one. In case of seed color, which color is dominant?
Yellow,
Yellow,
Yellow. Seed color yellow is dominant. Okay. Flower color, which one is dominant?
violet,
violet,
Violet.
Then seed shape,
round, round,
round,
pot color,
green,
green.
Green.
green is dominant.
yes,
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 sir.
sir.
Pot shape
full or inflated,
Inflated,
Full.
as a full or inflated. Flower position
Axial.
axial,
and stem height.
tall,
Tall.
So these are dominant characters. Very good. Same thing if I write for recessive, just tell me faster. Seed,
green,
seed color green,
green,
flower
white,
color
white,
next,
wrinkled,
wrinkled,
Wrinkled.
wrinkled, yellow,
yellow,
Yellow.
constricted,
constricted,
Constricted.
terminal,
short, dwarf,
terminal and
Dwarf. Dwarf.
dwarf. So these are different characters. What are these are traits actually? These are traits. So these traits are recessive traits. I have given definition of homozygous, heterozygous.
Yes, sir.
Hmm?
Yes, sir.
Allele, what is the definition of allele? Tell me.
Alternative genes
Alternative forms of gene which are present
homologous
at the same.
chromosome at same locus.
Yes, Ria.
The alternative forms a gene which are present at the same locus of two homologous chromosomes.
Okay, so these are alternative
Forms
of
Gene.
a gene. Okay, alternative form of a gene. It is the same gene, its alternative form. Okay, alternative form of a gene which is found
At same locus.
at the same locus, hmm,
Of
locus
two homologous
of
chromosomes.
two homologous chromosomes. So that is allele, right? Okay, very good. Now we know allele. Tell me, one normal person can have how many alleles for a gene?
Two.
Is it possible that more than two alleles are present?
No, sir.
Yes, Ambika, Anupriya, Vishnu. Is it possible that one person can have more than two alleles of a gene? This is one chromosome. Okay. Now this is homologous chromosome. Now check it here. One gene present here. We are saying this is A, capital A, this is small a. In this position, another gene is present, capital B, and this is small b. Capital A and small a are alleles or not?
Yes, sir.
Capital B,
Yes, sir.
small b are alleles or not?
Yes, sir.
Yes, sir.
If we find that A is producing a gene product, same gene product is also produced by B. Same product. Say A is responsible for tallness, B is also responsible for tallness. Consider. Under that condition, capital A and small b are alleles or not?
No, sir. No,
Hmm?
sir.
No, sir.
No? Yes, Akhiruz, unmute, unmute.
Yes, sir.
So, capital A, small b are alleles or not?
No, sir.
Why not? Why not? Wait, Aziz, Devojit,
Yes, sir.
A and B, capital A and small b are alleles or not? Hmm?
Yes, sir.
Allele. But everybody is saying it is not allele. Yes, explain. Yes, Divakar.
Sir,
Hmm? Divakar, capital A, small b are alleles or not? Faster. No. Okay. Yes, Samili.
the position same नहीं है.
Because their positions are not same. Very good. So, because it is written here, the locus at the same locus, are these two are same locus, are not at the same locus. Is this clear? Devojit, is that clear?
Yes, sir. Yes.
Capital S, small b are not positioned at the same position. And that's why they are not allele. So, and though they are producing the same gene product, yet we will not consider them as same alleles. Okay, or alleles of each other. So capital S, small a are a single set of gene. Capital B are another set of genes. Is that clear? So LL concept, if it is clear, then let us go for
Sir,
hmm,
so why did Mendel's experiment for lub bean fail?
acha. Mendel has worked on how quick and lab beans, but in these two plants, pure lines were not found. Pure lines were absent, right? So if pure lines are absent, how can you go for? Actually, in these two plants, parthenogenesis takes place. Parthenogenetically, seeds are formed. Okay, so automatically, most of the plants were haploid. If parthenogenesis is taking place, that means what? From egg, they are getting a new plant. So in that case, plants will be haploid. If it is a haploid plant, can you find the homozygous condition?
No, sir.
No. So that was the problem with these two plants. So you can simply write that a pure lines were absent in these two particular plants at that time.
Okay, sir.
Achha. Now check what has happened. Mendel, so talented this person was, he has got some result, and because he was not able to match this, so what he has done, he has uh, formulated and he has actually given this concept. What is the concept? If some plants are tall, why they are tall? Because they have two factors. They must have two factors, otherwise it will not match. So this is his concept. For dwarf plant also, what he has said, similarly, there should be two factors. And now, next what he is saying, this capital T and capital T, they will get segregated, they get separated like this. Why? Because during gamete formation, there will be meiosis. See, when he has said all these things, at that time, meiosis was not being discovered even. Okay, see, that's why we are saying he was actually genius. Small t and small t, they must segregate in this way, okay, because they will undergo meiosis. So without knowing whether actually what meiosis is or whether meiosis is occurring or not, just to match his mathematical deduction, he has said that they must segregate like this. And in the next generation, during fertilization, these two will unite. Okay, these two will unite in this pattern. I'm most probably I have explained this in the last class. Have I?
Yes, sir.
Yes.
Yes,
Yes,
sir.
sir.
But still I'm explaining so that you should keep it in your mind that this was a very important step, and for you also this is very important. This pattern, how capital T and small t they are uniting, this pattern what he has discovered at that time, not discovered actually, he has formulated it that it should go like this, otherwise it will not match. So this pattern of inheritance is called Mendelian pattern of inheritance, right? This is called Mendelian pattern of inheritance. Another point you have to consider here, yellow, seed color is yellow, is dominant. And you see, pot color yellow is recessive. Okay, so remember this point. These 2 points you have to remember. Now, let us go for dihybrid cross. When we say dihybrid cross, what does it mean?
So two pairs of contrasting
Are considered.
characters.
In a hybridization program, in a hybridization program, if two pairs of contrasting characters are chosen, it is called a dihybrid cross. Chosen or considered, let's say. It is called a dihybrid cross. Now see, when it is a dihybrid cross, that means we are considering two sets of means characters. For example, yellow round seed. So how many characters? Yellow seed, round seed. Two characters. And green wrinkled seed. So here also two characters we are considering. For yellow, he has considered, say it is YY, and for round it is RR. If I write YYRR, will it be wrong?
Yes, sir.
Why?
No.
So Y should be capital
Why?
because Y and round both are dominant traits.
Where it is written that dominant trait has to be considered in capital, it is not written anywhere. It is when you are performing a cross, you don't know before performing a cross, you don't know whether it is. एक मिनट रुको, ऑफिस से फोन आया. हेलो.
Yes, sir.
Yes, sir.
कहाँ चले हम लोग?
So capital or
Acha,
small?
capital Y. So that before you perform a cross, you are not knowing, no, that whether it is a dominant character or a recessive character. So if you consider this small y, smaller as dominant, then also it is not wrong. Remember this point. So when we consider as a convention, we consider capital Y is dominant, capital R is dominant, right? And small y, smaller r recessive. But in exam hall, if we find that one particular cross is given, and in case you find that with capital Y considering yellow with capital Y, it is not matching, then also consider yellow with small y. Once this tricky question is been asked long back, I have forgotten the year, uh, not in AIMS or in a state board, this question has been asked. I've forgotten exactly when it is, but this has been asked, remember. And in this tricky way it is given, it matches only if you consider that characters which are in small letter are being considered as dominant. Is this clear? Saumili, is that clear?
Yes, sir.
So there is nothing wrong if we consider them as what? The small y as dominant. But as a convention, we use capital Y as dominant, capital R as dominant. Remember, and in exam hall, don't do this mistake. उसमें उस्तादी करके ना small y नहीं बिठाना, क्योंकि सब लोग ये अलाउ नहीं करेंगे. समझ में आया?
Yes, sir.
So I can consider capital Y, capital R as dominant, and small y, small r is also as dominant. Now tell me how many types of gametes will be produced from this genotype. Capital Y, capital Y, capital R, capital R.
2.
Which two?
YR, YR.
YR, YR means capital Y, capital R, and
Capital Y, capital R.
are the two. They're the same. How many types? Type will be same only.
1. Yes, sir.
One.
Hmm. So in this case, how many different types you can have?
1.
One.
Small y, small y. I think this is clear.
Yes, sir.
Only one type, no? Type is only one. Okay. उसमें फर्दर हम लोग जाएंगे उसके पहले और कुछ प्रैक्टिस करते हैं स्टेप बाय स्टेप चलते हैं कोई प्रॉब्लम नहीं चलो उसको बॉक्स में डाल दें Can you tell me how many different gametes will be produced from this genotype? Devjit, fast reply. Vishnu, unmute yourself. Are you attending the class? If you don't unmute, then it will be reported. Devjit. देखो चोरी पकड़ा गया
Yes, sir.
आयन हाँ बोलो तो इसमें कितना सारा गेमेट्स होगा How many different types of gametes will be produced? Okay, Ayan. They will get separated, no? So when gametes will be formed, then one of them will go to one gamete, another of them will go to another gamete. Is this clear? समझ में आया बेटा? Check it here. Capital A, small a. Is this clear?
Yes, sir.
Oi. Devjit, is that clear?
Yes.
Why don't we reply? Whenever I ask, promptly you have to reply. This is compulsory. Everybody is replying, they are not getting any problem. Then what is the problem with you? You have to reply immediately. Check it here. नहीं तो मम्मी को रिपोर्ट होगा रुको. So they will form gametes like this. Now check it here. If I say capital A, capital B, small b. How many different types of gametes will be produced?
4.
Yes, very good. Ambika, how many different types of gametes will be produced? Batao.
4.
4. Can you tell me which 4? Capital A. Will it be like this? Like this? Astha? Will it be like this? हाँ समझ बताओ.
Sir, capital A, capital B, small a, capital B.
Means not like this.
No, sir.
Okay. Capital A, capital B, capital A, small b, small a,
Capital
capital B, and small a,
small.
small b. Okay, very good. So it will form like this. Is this clear to all of you?
Yes, sir.
Okay.
Yes, sir.
So when gametes are formed, you have to make different sets. How many different sets are formed? Okay, Vishal. Now check it here. Capital A, small a, capital B, small b, capital C, small c. How many different types of gametes will be produced? Types. सिर्फ नाम बताओ. Total.
9.
9.
8. Sir, 8.
8.
Sir.
Will it be 8 or 6?
Sir, 8.
Confirm.
Yes, sir.
Anu,
Sir.
it will be 8 or 6?
Sir, 6.
Acha, which 6? Tell me. First will be
Sir,
capital A,
capital A, capital B,
then
capital
capital A, capital B, or capital A, capital B, capital C also.
yes, sir.
Okay. Next, second one. Capital A, capital B, small c, can I write? Yes?
Yes.
Acha. If you are having complexities, let me explain in a different way. One person has two buildings. Okay? Okay, two buildings. Right? Two car. थोड़ा टूटा फूटा कार हो गया है. Two car. Right? And being botany people, we must have a tree. Two coconut trees are also there. Right? Now he want to donate all these things to his two son. How will he donate? Hello?
Pick one. Pick
Hmm?
one from the sun.
Take one from everybody's, no? That means one house, one car, and one tree, right? And the problem is that all are different. Out of these two house, one is big, one is small. Out of two car, one is a bit costlier, and another is a bit cheaper. Out of these two tree, one is mango tree, one is coconut tree. Now, what should be the distribution pattern? He can choose randomly. So how many different combinations are possible? That is the thing what we are discussing here. So in place of house, car and tree, if we simply write
30 हो जाएगा.
हाँ 30 हो जाएगा, तुम कहाँ से निकले? अभी देखो, कैपिटल ए स्मॉल ए, कैपिटल बी स्मॉल बी, capital C small c. Anu, is that clear? So how many different combinations are possible? One combination is that capital A, capital B, capital C. This is one combination. Another is small a, small b, small c. Right? This is one combination. Another combination, what we can write? Capital A, capital B, and small c. And small a, capital B, small a. Acha. So we can write no? Capital A, capital B, small c. He can choose capital A, small b, capital C also. Right?
Yes, sir.
Yes, sir.
Okay. He can choose small a, capital B,
Capital A,
capital C.
capital C.
Okay. He may choose small a, small b, capital C. Hmm? Or he may choose what? Small a, capital B, small c. He may choose capital A, small b, small c. Or last one is this. So how many different possibilities are there?
8.
8.
8. So
तो
is this
स्मॉल ए
clear?
कैपिटल की स्मॉल सी से 2 बार लिखा.
हाँ, कहाँ पे रे? Small a, capital B, capital C. कहाँ पे दोबारा लिखा है?
तो स्मॉल ए कैपिटल बी स्मॉल सी.
Small a, capital B, small c. और कहाँ लिखा है?
Sir, ठीक ही I guess.
Sir, नहीं लिखा.
सर नहीं लिखा है.
Yes, sir.
तुम चश्मा पहनो रे. देखो हो गया ना? No problem. Is it clear?
Yes, sir.
So
Yes,
how
sir.
many different types?
8.
8.
8 different types are possible. Okay. Now I have another question. Capital A, small a, capital B, small c, small c. capital D, small d. How many different types of gametes will be produced? जल्दी बताओ.
Sir, 4.
Only 4?
Yes, sir.
Why it is so?
क्योंकि कैपिटल बी और स्मॉल सी सेम है.
Same means homozygous है.
Sir.
Yes, very good. Here you check 2 to the power 1 I can write. In this case, I can write 2 to the power 2. In this case, I can write 2 to the power 3. Yes. Now, in this case, what is the problem? These two are in homozygous condition. So, the formula is actually 2 to the power n. But thing is that the n includes only the heterozygotes. Now, to make it simpler, let us make capital A, capital B, small c, capital B. This is one gamete, right?
Yes, sir.
Yes.
Another gamete, small a, capital B, small c, capital D. I can write.
Yes, sir.
Another gamete will be capital A, capital B, small c, capital D. And another,
Wait,
uh,
it is same.
Small d.
Small d.
small d, exactly, small d. And what? Small a,
Small
Small a,
capital
small
B,
b, small c,
Capital
small c.
small c,
small c,
small b
small
नहीं
d.
है, capital B. Small a, capital B, small c, small d.
Small d.
Can I write?
Yes, sir.
Can I write
Yes.
any
Yes,
more?
sir.
Any more gametes can I write?
No, sir.
No, sir.
Is it clear to all of you?
Yes, sir.
So under that condition, what will happen? Mm, we are not considering the homozygous. Okay, the gametes which are in homozygous condition, we are not to consider. So जल्दी जल्दी इसका बताओ तो. Say small a, capital B, small b, small c, capital D, capital. अच्छा उल्टा हो गया. Capital C, small d, capital E, small e, capital F, small f. So how many different types of gametes will be produced?
8.
8.
Is it clear?
Yes, sir.
Yes, sir.
So you have to choose this one, and last thing. So 8. If you were asked, if we make a cross between capital small a, capital B, small b, with capital A, same genotype, say capital A, capital B, small b, how many different zygotes will be formed?
Four.
16. Sir, 16.
Achha, check it here.
4.
Total how many zygotes will be formed? How many zygotes will be formed? So you check it here. This is 8. Sorry, this is 4. It will produce 4 different types of gametes, right? Simply you check. It will also produce how many?
4.
4,
4. 4 into 4. So answer will be 16.
16.
You have to consider this formula. Is it clear?
Yes,
Yes.
sir.
Capital A, small a, capital B, capital C, small c, is crossed with small a, capital B, small b, capital C, small c. Now tell me how many different zygotes will be formed.
16.
16.
16.
Is it 16 even in this case?
32.
32. How? How many different gametes will be produced here?
4.
4.
2 to the power 2, no? So 4.
4.
In this case, 2 to the power
4.
2, so 4. So 4 into 4, total number is 16. Yes. I think this is clear to all of you. So this is how we calculate the total number of gametes, total number of zygotes. Questions you will find, this type of questions will be given to you. Right? Can you
तो
handle?
उसमें 4 कैसे
इसमें रे
तो वो कैपिटल ए स्मॉल ए कैपिटल बी कैपिटल सी स्मॉल सी।
इसमें कितना होना चाहिए? रणवीर,
पांच
कितना होना
नहीं
चाहिए?
होगा क्या?
5। यहाँ पे देखो, तो यहाँ पे we have to consider only heterozygotes. Check it. Capital small a and capital D small d. You have considered. Have you considered this too?
ओ सर सॉरी सर वो कंफ्यूज है।
इसलिए तो कंफ्यूज अभी होना जरूरी है। अभी कंफ्यूज हो जाओ, कोई प्रॉब्लम नहीं है। एग्जाम हॉल में कंफ्यूज मत होना। सो इज दिस क्लियर? सो इसमें 5 बहुत लोग बोलेंगे ये 5 होना चाहिए ना, कैसे 5 हो जाएगा? अभी तुम गेमेट्स निकाल के देखो ना, ठीक है?
यस सर।
इसलिए तो मैं इतना डिटेल्स में जाके समझा रहा हूँ ताकि इसमें कंफ्यूजन ना हो। ओवर।
Yes, sir.
Yes, sir.
So I think we can handle this question. Now let us go for dihybrid cross. इसमें नेक्स्ट जनरेशन क्या होगा? कैपिटल Y, स्मॉल y, कैपिटल R, स्मॉल r. When these two gametes unite, it will produce this result. Okay. Now, how many different types of gametes will be produced by this F1?
4.
Which 4?
Capital Y, capital R, small y, capital R, small y, small r.
Capital Y, capital R. Next, you say capital Y, small r, right? So both capital will go. Then small y, capital R, and small y, small r. Easy? Now, in this case, if we go for selfing, so these are gametes. This is parental generation. These are gametes. This is F1, first filial generation. These are gametes. So it will be a bit difficult for us to make cross directly. So we will place it in a table. That table is known as Punnett square problem. Why it's called Punnett square? Punnett first time has used this type of table. That's why it's called Punnett square table. So in Punnett square, if we use, तो बनाना पड़ेगा। तुम लोग भी बनाओ, मैं भी बनाता हूँ। One. There will be another one. Okay. ज़्यादा बड़ा हो गया। Now check. We can place, say, once this was the question, in which side we use male gametes? Is it a question? You can put male gametes in this side, you can put male gamete in this side. Absolutely, it depends on your intention. Now see, that was the question, because in NCERT, they use male gamete on the upper side, female gamete in the lower side, right? And holding this, they are asking you this question. See this level of question. Hmm? Capital Y, capital R. Capital Y, small r. Small y, capital R. Small y, small r. We can write. It will be what? Capital Y, capital R. Then capital Y, small r. Small y, capital R. Small y, small r. Right? You do one thing, you give box number. See, this is box number 1 in the corner. You just write the box number. This is box number 2, box number 3, box number 4. This is 5, 6, 7, 8, right? 9, 10, 11, 12, 13, 14, 15, and 16. You place the data. You place the data and let me color it. Are you placing?
Yes, sir.
Okay, place the data. मैं इसको सुंदर बनाता हूँ, तुम लोग काम करो. Have you written capital Y, capital Y, capital R, capital R?
Yes, yes, sir.
This is capital Y, capital I, capital R, small r.
Yes, sir.
Is it completed? Have you written this? Chart
Yes,
is
sir.
completed.
Yes.
You'll find capital Y, capital R, small r. You can place it here also. It will be capital Y, small r, small r. And it will be small y, capital R, capital R.
Sir, the third one is wrong.
Which
Means
one?
capital R, capital R.
Yes, yes. Thank you. Capital R, small y, capital R, capital R. Right? This is also, it will be what? Capital Y, small r. This is small y, capital R, capital R. It will be small y, small r. It will be small y, capital R. It will also be the same. It will be what? Capital Y, small r, right? It will be also the same. And these are capital Y, capital R, small r. All the blue will be of the same color. Matching?
Yes, sir.
Yes, sir.
Now you do one thing. Below you write how many you have got yellow round. And yellow wrinkled, green round and green wrinkled. Now check it here. Number 1, 2, 3, 4, 5, 6, 7, 9,
10,
10, and
13,
13. So how many
9.
altogether?
9.
9. Okay. Yellow wrinkled. We'll find 6, 8, and 14. Right?
Yes, sir.
3. And what is left? Green round. 11, 12,
11 to 15.
and 15. Right? So altogether 3. And
Sir,
16 is, yes.
sir, you have repeated 6 in so yellow round में 6 नहीं होना चाहिए था.
ओ अच्छा अच्छा अच्छा, ये 6 कहाँ से आ गया? 7 होगा, है कि नहीं? 5, 7, 9, 10, 13. Yes. 5, 7, 9, 10, 13. Okay. So in the last one, what you can say? Only 1. So the ratio, this is the phenotypic ratio. What you can observe? How many? 9:3:3:1, right? So phenotypic ratio is coming as 9:3:3:1. Then what will be the genotypic ratio?
Sir, 1 is to 2 is to 1 is to 2 is to 4 is to 2 is
Yeah.
to 1 is to 2 is to 1.
ऐसे नहीं बोलते. 1:2:1 is to 2:4 is to 2:4 is to 2 is to 1 is to 2 is to 1. तुमने यही बोला ना? अब मैं भी जानता हूँ तुमने यही बोला, लेकिन ऐसे नहीं बोलते. इसको कैसे बोलोगे? 1:2:1. This is the monohybrid cross ratio. Make it double. 2:4:2 is to 1:2:1. Right?
Yes, sir.
Okay. So if you read in that way, then you will not miss this. 1:2:1. Make it double. 2:4:2. Then 1:2:1. Right? So in this way, if you read, then you will never miss. Check it here in the graph. It is only 1. So 1, these two are same, and this is 1. So 1:2:1, right? These two green colors are same, all the blue are same, and these pink colors are same. So this is what? 2:4:2 we can say. This is 1:2:1. So what is the ratio coming? 1:2:1 is to 2:4:2 is to 1:2:1. Is it matching? Hello?
Sir.
Yes, sir.
So if you are being asked that what is the dihybrid ratio, see why I am putting stress here. You may get a question where the ratio of genotypic ratio will be given, say 1:1:1 is to 2:4 is to 2 is to say 2:1. So like this one ratio you have got. Now is it correct? 4 different types of ratio will be there. Randomly will be given. If you know your ratio, then how will you go? You'll go on cutting. 1:2:1 is to 2:4 is to 2 is to 1:2 is missing. Say it is given 2 here. 2:1. So you can go on cutting and you can understand, okay, which one is your and which one is not. Is that clear? So in this way, you can find your answer very easily. And this is not tough even to remember. Say how to make, say we can deduce it from monohybrid table. Let me show it here. Wait a minute. Lia Gs. Check it here. What is the monohybrid ratio? I can write monohybrid was 3:1. 3:1 square if you write. Next, what you can write?
-1.
You can write like this. Next, we can write 3 into 3:1 is to 1 into 3:1. Then we can find what? 9 is to 3 is to 1. Can we obtain it from the monohybrid ratio? Yes. Same thing for dihybrid genotypic ratio. How can we obtain? What is the ratio?
1:2:1.
1:2:1. Square? Okay. 1 into, I can write, is to 2 into, I can write, is to 1 into 1 is to 2. Then what will be the ratio coming? Check. 1:2:1. Is to 2:4 is to 2. It will be double. And again 1:2:1. Is it matching?
Yes, sir.
Yes, sir.
If you are asked, what is the trihybrid genotypic ratio? Tanvir, batao jaldi.
Sir,
Trihybrid genotypic ratio.
ट्राइहाइब्रिड इसके साथ मोनोहाइब्रिड का
क्रॉस करो तो क्या होगा?
ट्राइहाइब्रिड
मैं बताऊं, 1:2:1:2:4:2:1:2:1 2:4:4:2:4:8:4:2:1:2:1 2:4:1 राइट?
सर क्या बोल रहा है सर?
देखो ना,
Oh my
एकदम
God.
आसान।
तो क्यूब करेंगे क्या?
क्यूब करने से तो मिल जाएगा ना। तो इसको, ये तो
Yes,
स्क्वायर
sir.
का रिजल्ट है, उसके साथ दूसरे बार और एक 1:2:1 का मल्टीप्लाई कर दो तो क्या होगा? ये आएगा पूरा 1:2:1:2:4:2:1:2:1 उसके
और
डबल
उसके
कर
बाद
दो।
2 के साथ मल्टीप्लाई करके आएगा।
हाँ, तो डबल हो जाएगा ना। तो 2:4:2
Yes.
4:8:4:2 again write the same thing 1:2:1 2:4:2 1:2. Is it clear?
यस सर।
करके देखो, बहुत मजेदार है। एलेक्स बताओ तो इसका रेशियो बताओ।
सर खत्म नहीं हुआ।
अच्छा चलो।
सर हो गया।
बोलो।
तो 1:2:1:2:4:4
अरे 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 ऐसे नहीं, ऐसे नहीं बोला ना, 3 3 करके उसको सेपरेट करो।
यस सर। 1:2:1 2:4:2 1:2:1
ओके
2:4:2 4:8
उसको रुको रुको ना, पूरा पूरा सेट को अभी डबल करना है ना, तो तुम्हें उसको बोलने में ऐसा बोलना है, देखो 1:2:1 2:4:2 1:2:1, then 2:4:2 4:8:4 2:4:2, right? Again 1:2:1 2:4:2 1:2:2। समझ में आया ना?
यस सर।
अभी बताओ एक बार।
1:2:1 2:4:2 1:2:1
Okay.
2:2 4:8:4 2:4:2 1:2:1 2:4:2 1:2.
Simple, very simple. Does it
Sir.
require any memorization?
No,
No, sir.
No, sir.
No, absolutely not. If you maintain this gap, 1:2:1, then 2:2 2:4:2 1:2:1. So very simple. Over.
sir.
Yes, sir.
हाँ.
सर ये
53.
phenotype में भी एप्लीकेबल होगा, मतलब ट्राइहाइब्रिड के लिए भी।
हाँ तो उसमें क्या होगा, ट्राइहाइब्रिड फेनोटाइपिक रेशियो अगर पूछेगा, इट विल बी 27:9:9:3:3:9:3 करके देखो। 27:9:9:3:9:3:1 मिला कि नहीं?
यस सर मिल गया।
And in trihybrid cross, how many zygotes will be formed?
8.
64 zygote.
Oh, yes sir.
How many? Gametes will be 8, zygotes will be
64.
okay.
64.
64. Is this clear?
Yes, sir.
Yes, sir.
Yes, sir.
तो डाइहाइब्रिड क्रॉस में देखो तो हम लोगों ने ओरिजिनली कितना कैरेक्टर से स्टार्ट किया था? येलो एंड राउंड क्रॉस्ड विद ग्रीन एंड wrinkled. That's all. And original characters are yellow round and green wrinkled. Now how many characters we have got? Yellow round, yellow wrinkled, green round, green wrinkled. So how many characters you have got?
4.
4. That means these two are what? Recombinants. These two are recombinant character. Is that clear?
Yes, sir.
So these two are recombinants. If you are asked that what is the ratio of the recombinants in dihybrid cross, what will be the answer?
1:1.
Yeah, 3:3. You can say
So
1:1. But in dihybrid cross, what is the ratio?
3:3.
Yes, it is 1:1. You can say, you can convert it. But original ratio is 9:3:3:1. No, so they may ask you only about this, and in option you will get this option 3:3. Is this clear?
Yes sir.
समझ में आया ना? क्योंकि यहाँ से डिराइव किया है ना, इसलिए उसको चेंज जनरली नहीं करते हैं, तो डायरेक्टली उसको रख देते हैं 3:3. You can say 1:1, but it will be 3:3 for this two. 3:3 is 1:1 you can consider, but in option you will not find 1:1, you will find 3:3. Tanbir,
Yes, sir.
asking something?
No, sir.
Achha. So what we can observe here, yellow character originally it was going with round, but now yellow character is going with wrinkled also. Green character originally it was going with wrinkled, Now it is going with round also. That means all the characters, they can independently assort. Are you getting my point? All the characters are independent, they can independently assort. So this is another very important point, and that's why Mendel has given his fourth postulate, or the third law. Which one? The independent
Law of
assortment.
independent.
Independent assortment. So this law of independent assortment is obtained after which cross? After this
Dihybrid cross.
dihybrid
Dihybrid
cross.
cross.
Okay, so remember this. First two laws have been described from which one? Monohybrid cross.
Monohybrid
And
cross.
third law is being derived from dihybrid cross.
Dihybrid cross.
याद रहेगा?
Yes, sir.
Yes,
Yes,
sir.
Simple, very simple. Up to this much. Now one thing I have not told you. S. Blixt, one scientist. What is the name? S. Blixt. कोई लिखने के लिए जगह नहीं है.
सर आपने नोट में दिया है.
दिया था क्या?
Yes, sir.
S. Blixt has given that the characters which are present where the different chromosome. How many chromosomes are involved?
4.
4 chromosomes are involved. Of which, in which chromosome maximum numbers of characters are present?
4.
Chromosome number 4. Which character is present in chromosome number 1?
Seed color and flower color.
And in chromosome number 7?
Pod color. No, sir. It was seed shape.
And in chromosome number 1? Oh, sorry, 5?
Seed. Pod color.
Pod color. Okay, and in rest are in 4. In chromosome number 4, which 4?
Stem height, then flower position, and
And
I forgot.
उसका देखना पीडीएफ में है ना उसका
Yes, sir.
पीडीएफ में मैंने दिया है उसका देख के रखना ठीक है so basically only one question which is very common that in which character Mendel has chosen maximum which chromosome he has chosen maximum character from which chromosome. So this is chromosome number 4. So another point, do you know what is linkage? Now come to another point. I'll describe this in the later portion again, but here you try to understand this. Now check it here. Say it is yellow and round. Consider yellow and round characters are present in the same chromosome. Consider this. Okay. And in F1 generation, it will be like this, small y and smaller. Is this clear?
Yes, sir.
Okay. Now check it here. When a cross will occur, during meiosis, what will happen? Here, during meiosis, capital Y, capital R can go together, right? Small y, small r also going together. And because what happens here? Crossing over takes place. During meiosis, crossing over takes place or not?
Yes, sir.
Yes, sir.
Abhishek, in which stage of meiosis crossing over take place?
So pectine.
Pectine, very good. In pectine, when crossing over take place, then exchange of segment take place.
Yeah.
If it is a chromosome like this, say two chromatids are also visible here, right? Under that condition, what you can find? चलो इसको ऐसे बोलो. Check it. It will also be capital Y, capital R, it will be small y, smaller, right? Now, if exchange of segment take place, then what we can say? This part of this chromosome will be exchanged with this one. So it will come here and it will appear here. Under that condition, what will happen? It will be small y, capital R or not?
Yes, sir.
Hello?
Yes, sir.
Yes. So these two
Yes.
will be recombinants. Small y, capital R and capital Y, small r will be recombinants or not? So these recombinants are formed due to crossing over, right?
Yes, sir.
Yes, sir.
सबको समझ में आ रहा है तनवीर? Is that clear, Vishal?
Yes, sir.
No, sir.
Not clear.
No,
If
sir.
crossing over take place here, then what will happen? Capital Y and small y, they will exchange their positions. We know crossing over. Yes or not?
No, sir.
Wait. If crossing over, you know, when crossing over take place, then exchange of segments of chromosome takes place. Is that clear, Vishal?
Okay, sir.
Okay. So when exchange of segments of chromosome take place, then what will happen? Capital Y from this position will go to this position. Small y will come to this position, right? So under that condition, what will happen? Capital Y is going now with small r. So this is a new combination. So this is a recombinant, small y and capital R. Now they will form another combinations, right? So what will happen? Under this condition, when they will form gametes, they may form capital Y, capital R. They may form capital Y, small r, as here. They may form small y, capital R, or they may form small y, small r, right? So 4 different combinations are possible or not? Yes. Is this clear? Ambika? Ankita, what happened? You are not replying today.
I'm there.
Okay. So is this clear now?
Yes, sir.
Vishal, is it clear now?
Yes, sir.
Okay. Now check it what? If crossing over take place, then only they will form recombinants. If crossing over do not take place, then what will happen? Recombinants will not be formed, right?
Yes, sir.
Okay,
Yes, sir.
recombinants will not be formed. Remember this point, very important point. If crossing over do not take place, then that condition is called linkage. You should know, if two characters are present in the same chromosome and sometimes they have a tendency, what? Not to go for crossing over. That means they want to stay together. So that tendency is called linkage. Under that condition, both the gametes will, both the factors or both the genes will go to the same gamete, right? Under that condition, recombinants will not be formed. Is this clear? If recombinants are not formed, then it will form link. It is due to linkage. If linkage is involved, then recombinants will not be formed. If recombinants are not formed, then these two characters will not appear, right? So if these two characters are not appearing, then Mendel could not formulate which one? Independent, the law of independent assortment. Is this clear? आया बात समझ में?
Yes, sir.
Is it clear to
Yes,
all of
sir.
you? Right? So that is why often you are being asked that if linkage were involved, then which law Mendel could not formulate? Answer will be law of independent
Hard law.
assortment. Okay, one point. Another point you may be asked in a different way. Say Mendel has chosen 14 characters. 7 characters means 14 traits, right? And in P plant, how many chromosomes are present? 14 chromosomes are present, means 7 pairs of chromosomes are present. Consider in P plant, instead of 14, there were 12 chromosome. They may ask you. If we consider there were 12 chromosomes, then what we can say? All the characters, what Mendel has chosen, all the 7 contrasting characters, they will be present. They will be more condensed, no? So there is a possibility that those characters what are present in different chromosome would present in the same chromosome. Are you getting my point? Those characters now are present in 14 different chromosomes. Okay, if number of chromosomes are less and same number of character if we consider, then there is possibility that the characters will be present where? In the same chromosome. Under that condition, there is possibilities of linkage. Is this clear?
So why? Sir,
Say,
link.
this is clear. This part is clear, I think.
Yes.
Okay. Now consider you have how many? You have altogether how many chromosomes? 12 chromosomes instead of 14. Say, consider you have these 3 chromosomes. And when you are having 3 chromosomes, you have chosen characters, 3 characters, okay, in which are present in 3 different chromosomes. Is this clear?
Yes, sir.
Considered like this. It is capital A, this is capital B, and this is capital C. Characters are present in this way. Now what has happened? If, what is the question? If number of chromosomes were less, say this pair is not present, but characters are remaining same, then there will be possibilities, no, that characters are present in the same chromosome.
Yes, sir.
Yes, if characters are present in same chromosome, then there is chances of getting mutation, sorry, linkage. There will be chances of having linkage. Is this clear? Linkage occurs when characters are present in the same chromosome. Are you getting my point? If characters are present in different chromosome, can it show linkage?
No.
No. So linkage is possible only when characters are present in same chromosome. So if number of chromosomes were less, then ultimately characters will be more condensed, you know, and they will be present in same chromosomes. So if more number of characters are present in same chromosome, then the probability of getting what? Linkage will be more. If probability of getting linkage is more, so probability of not getting the law of independent assortment is also more, right?
Yes.
So that is the point. So not, they are not directly asking you, but they may ask you in this way also. Is this clear?
Sir,
Hmm.
sir, if characters are present on the same chromosome, then why is linkage like more? That's what I didn't understand.
Acha, forget everything. Just tell me one point. If characters are present, say one character A and B, they are present in A and B, they are present in different chromosome. Yes.
Yes, sir.
Can A and B show linkage?
So in less probability.
Uh-huh.
So less probability.
Less probability. Linkage means what? That means capital A and capital B, if every time go to the same chromosome, is there any reason that A and B will go every time to the same chromosome? Is there any reason? Isn't it just tossing a coin? If you toss a coin, say few thousand times, what is the probability to get the
50-50.
50-50? Is that clear? So it will not show linkage under that condition. Small error, what is coming, that will be called as sampling error. Okay, small error, what will become as, that will be called as, so don't say that they will show linkage, they will never show linkage. So linkage will be shown only when characters are present, say A and B are present in this chromosome. If crossing over takes place at this position, then only they will get changed. If crossing over is taking place here or anywhere else, then they will show linkage only. Is this clear? So probability to have linkage will be more if characters are present in same chromosome. Is this clear now?
Yes, sir.
That will be even more clearer when we discuss linkage thoroughly. Okay, time is up. So we have discussed today Mendel's dihybrid cross. We have also discussed how to produce different types of gametes, how to calculate total number of zygotes, okay, and some more points we'll discuss. In the next class, we'll discuss about test cross, back cross. So if I ask what to start, you just say we have to start what?
Yes,
Back
cross.
cross, test cross. Okay then. Thank you everybody.
Thank
Thank
you,
you,
Thank
sir.
you,
You,
sir.
sir.
Have you written all these things, whatever we have discussed today?
Yes, sir.
Yes. So I will not share any PDF today. Will it do?
Yes, sir.
Divakar, is that clear?
Yes, sir.
Okay. I think you have written everything, and this part already you have got it in your in earlier PDF you have already got. So you just go thoroughly. I think you have done this particular one.
Yes, sir.
So किसी ने अगर cheating किया तो उसको नहीं मिलेगा. Okay then. Thank you.
Thank you,
Have a
sir.
Thank
good
you,
day.
sir.
Have a good day.
Sir.